Hello everyone, I'm Nirmiti Nimbalkar. I'm doing PhD under the able guidance of Dr. R.P. Chondekar, Associate Professor, Department of Zoology. This is the presentation regarding my overall research work, which is entitled Ecobiology of Freshwater Bryozoa and Sponges of Kagzipura and Mombata Reservoir. I've selected the topic because the freshwater bryozoa and sponges are neglected invertebrate group in India. In India, pioneering work is carried out by Anandali and Rao, and they have given the distribution of bryozoa, which is restricted to central and eastern part of India. Sponges, out of 4,500 species of sponge, mostly are marine. Only single family, the Spongelidae, being freshwater. Around 100 freshwater species are reported, of which 31 are found in India. After the work initiated by Anandali, Peni, Chaturvedi, Tonpi, Yeragi, very little work was done to understand sponges and its ecological significance. Much of this region is still unexplored for bryozoa and sponges. Data on association of bryozoa and sponge is restricted to marine ecosystem only, and in freshwater ecosystem, the exact affiliation between them is not yet clear. So, my idea about this research work is to explore the more data about this neglected invertebrate in given study area, to add valuable data in freshwater fauna of the given area by studying the diversity of freshwater bryozoa and sponge, by studying the morphology of the colonies and fruiting bodies. Then to understand the real community structure of limnitic ecosystem and their interdependence by studying the interaction between bryozoa and sponge and their interaction with the other organisms also. Then to find out the role of freshwater bryozoa and sponge in ecosystem and their applicability in changing ecosystem. So the methodology will include collection of sample which will be done from the study area, that is Kagzipura and Mombata Reservoir, for 1 year. Regular sampling will be done twice in a month at the interval of 15 days. Different kind of submerged substratum will be collected in labeled sample bottles. Similarly, water sample will be collected to check the chemical and biological water parameters. To check biological parameters of water, plankton sample will be collected with the help of plankton net. Collected samples will be analyzed under dissecting microscope and images will be captured with the help of eyepiece camera. And then identification of species will be done by having scanning electron microscopic images of fruiting bodies and preserve samples by using standard taxonomical keys. So the targeted outcome of my research work is it will facilitate the phylogenetic reconstruction of species position based on molecular data for both bryozoa as well as sponge. In addition, non-molecular characters, example morphological or paleontological, can be mapped on the molecular tree serving as a backbone. Thus, molecular and morphological data complement each other and are both equally needed to understand the evolution and presence of these organisms in ecosystem that I'm planning to study. So, thank you.